Ladies and gentlemen, depending upon where you're from, myself is Muhammad Syam Akram, the founder president of Poultry Professional Society. So I warmly welcome you all to our webinar number 4 of the Society Webinar Series 2025. As you know, PPS is a dynamic professional platform uniting scientists, educators, industry experts, and students with a shared mission to advance innovation, knowledge exchange, and collaboration for the sustainable growth of the poultry sector. So once again, I welcome you all to our webinar number 4 and thank you very much for joining. Today, we are honored to host a world-renowned virologist and poultry health expert, Professor Dr. Daniel Perez, Professor of Poultry Medicine, University of Georgia, USA. So let me give a very brief introduction of Professor Dr. Daniel Perez. Dr. Perez is a Georgia Research Alliance Distinguished Investigator and holds the Caspel S. Edson Chair in Poultry Medicine at the Poultry Diagnostic and Research Center, University of Georgia. With more than 2 decades of pioneering research, he has advanced global understanding of avian influenza, its molecular determinant of host adaptation, and the development of innovative vaccine strategies. His landmark work on quail as an influenza reservoir influenced regulatory changes in Hong Kong poultry markets in 2002, while his US leadership in the USDA-funded prevention and control of avian influenza program built new framework for research and surveillance. His laboratory is also part of the NIAID-funded CEIRS network, contributing to preparedness against zoonotic and pandemic influenza. In recognition of his scientific leadership, Dr. Perez has served on NIH, USDA, and CDC evaluation panels and continues to guide policy and practice in poultry health globally. So today we will share with us his expertise on the topic innovations in poultry vaccination, live attenuated strategies against bird flu, and the topic which is shown over the screen, wings of worry, avian influenza viruses. So with that, I'm delighted to invite our respected session chair, Dr. Nasir Mukhtar, who is our Director of Poultry Advisory Committee, PPS, to deliver his welcome note. Please, over to you, Dr. Nasir Mukhtar. Thank you very much for joining.
Thank you very much, Mr. Syam Akram. Ladies and gentlemen, and welcome to the Dr. Daniel Perez. So let's start our today's seminar. It's my great pleasure and privilege to welcome you all on behalf of the Poultry Professional Society to this highly significant session. We extend our deepest gratitude to the Professor Dr. Daniel Perez for joining us. Your contribution in influenza virology and vaccine development are of the immense globally impact, and we are eager to learn from your insight. I also acknowledge the commitment of the Poultry Professional Society and its leadership in creating this vibrant platform for knowledge sharing, which connects industry, academia, and research. So please welcome Dr. Daniel Perez.
Thank you very much, Dr. Nasir Mukhtar, for such a wonderful welcome note to our speaker and to the participants who are attending this session. So without further delay, it's over to you, Dr. Daniel Perez, and thank you very much for joining us.
Thank you, everyone. Thank you for the opportunity to present some of the work that we do today, and to the audience for taking the time. I know it's late in the day for some of you, so I truly appreciate that you take the time to hear what I might be able to tell you in the next few minutes. So before I speak about the work that we've done in the lab, I want to acknowledge the people that really made it happen. We have a large group of collaborators in the US and elsewhere in the world. But those highlighted in bold here are the people that actually play a major role in the work that I will be presenting today. So I want to acknowledge their contribution first and foremost. So I divided talk to talk about a little bit about bird flu, something that you probably know a lot about, why we care about zoonotic transmission of avian influenza viruses, some of the work that we've done with this new class of modified live attenuated virus vaccines that have the potential to be applied through mass vaccination strategies, and the little work that we've done with these vaccines also to see whether they would work against H5N1, highly pathogenic avian influenza. I will briefly touch upon an aerosol exposure system, but with a focus on what we've done for delivery of the vaccine. And I realize that this slide is a little bit wrong. I will not be talking about the Jersey cows. I will be talking about something else related to diagnostics. I apologize for the mishap here. This slide was confused. I mean, it was put there by mistake. So today we'll be talking about influenza viruses, more specifically about type A influenza viruses or flu AB. As you probably, many of you know, these are viruses with a lipid bilayer envelope that is derived from the host plasma membrane. These are pleomorphic particles. That means that they can adopt many different shapes. Sometimes sort of like a bullet shape. Sometimes these round shapes. But essentially, the virus is very malleable. It's very plastic in terms of the morphology. The genome of the virus is of RNA, is segmented, negative sense. So that means that the RNA themselves are not infectious. And it contains an RNA-dependent RNA polymerase complex. I mentioned that the most important members of this family are the type A influenza viruses, and there are many reasons for that. One of the reasons is that on the surface of these viruses, there are two major glycoproteins known as hemagglutinin and neuraminidase, and there is a large diversity of these HAs and NAs in terms of their antigenic makeup. So based on their antigenic makeup, these viruses are divided into subtypes. And so far, we have identified 17 HA subtypes and 9 NA subtypes. And I put there in parentheses avian because those are being found in avian species. There are additional HA and NA subtypes that have been found in fruit bats, but those are slightly different viruses. And I will not be talking about them that much. As I mentioned, the virus contains 8 segments of viral RNA, and on the surface, we have these 2 sulfate glycoproteins that also undergo small genetic changes, more mutations. This is because the polymerase complex of influenza viruses cannot correct mistakes. The polymerase is prone to incorporate mistakes or to make mistakes that cannot fix, and that further increases the diversity of these viruses in nature by a process known as antigenic drift. Because of the 8 segments of RNA, the virus can also, and it's very easy to imagine, exchange in segments. So when 2 or more, influenza viruses infect the same host at the same time, there is plenty of opportunities for gene segments to be exchanged between strains, and that actually contributes to furthering the diversity of these viruses in nature. When it comes to the host range of these viruses, type A influenza viruses have a natural reservoir in wild aquatic birds of the world. That's where all the HA subtypes have been identified. I clarify again, the exception of few of those have been found in fruit bats. But in these birds, these viruses don't seem to be causing much clinical disease. They replicate in the gastrointestinal tract most of the time. There is some respiratory involvement, but most of the time the infection is in the gastrointestinal tract. The virus is excreted through the feces into the water and the virus can perpetuate a cycle of fecal-oral transmission in this natural reservoir. From this natural reservoir, occasionally, and because the virus can mutate, generates a strain that can jump to other species. And galliformes, the subject of today's talk, are perhaps the lowest barrier for these viruses to jump to. Galliformes are not natural cause of avian influenza. The natural cause are, as I mentioned, wild aquatic birds in the order Anseriformes and Charadriformes. Galliformes cannot and should not be considered natural cause of avian influenza. And once a virus jumps into Galliformes, you can expect further adaptation in these viruses. And many times these adaptations lead to better ability of these viruses to replicate in the respiratory tract of the birds, and the accumulation of additional mutations that can make jumping into other hosts, for instance, humans, more effectively. And we have many examples of those. The subtypes H5, H7, and H9 are clear examples of viruses that have really emerged through poultry and have shown the ability to infect humans more effectively than those that circulate in wild birds. A key component in the interspecies transmission of influenza viruses is the receptor specificity of these viruses. The hemagglutinin of these influenza viruses is in the form of a homotrimer, so 3 monomers of HA. On top of the HA, there is a globular head, with each monomer having a shallow pocket known as the receptor binding site, is highlighted in red there on the left of the figure, and a stem that basically holds that globular head in place. The receptor binding site typically recognize sialic acid receptors. These are terminal sugars on glycoproteins or glycolipids on the surface of the cell. Viruses that circulate in wild birds tend to recognize these sialic acids as an alpha-2,3 conformation. And eventually, when these viruses jump to other species, they may switch that receptor specificity to an alpha-2,6 receptor specificity. And this is the kind of receptor specificity that is known to allow human influenza viruses that at one point in time were part of the avian reservoir to eventually become adapted to humans. Okay, so but today we're talking mostly about this jump from wild birds to galliformes, and we'll focus the attention mostly on the galliformes. So in galliformes, avian influenza or bird flu, it is a threat to birds and the economy. It was originally identified as fowl plague in 1878 in Italy. Since then, we have had many outbreaks of avian influenza, particularly of those that are associated with deaths or having to call the birds just to control the infections. Since 1981, there has been the adoption of a nomenclature or a terminology for avian influenza viruses that divide them into either low pathogenic avian influenza viruses or highly pathogenic avian influenza viruses. The low pathogenic avian influenza viruses are essentially viruses that belong to any subtype of avian influenza, but I would say that the most prominent among these viruses are the H9 and 2 because of their geographic distribution around the world, something we're going to discuss in a few minutes. These low pathogenic avian influenza viruses cannot be confused as something that is mild. Usually they can result in significant problems to the poultry industry by outbreaks of respiratory disease that leads to drops in egg production, or associated with high mortality in the face of secondary bacterial infections that are promoted by this initial infection with the low path viruses. In the case of the highly pathogenic avian influenza viruses, those are restricted to 2 subtypes, H5 and H7s. They become highly pathogenic in chickens, usually, or in galliformes, because their HAs tend to accumulate mutations in an area known as the cleavage site. And by doing so, the virus accumulates a series of basic amino acids shown in the cartoon there on the right. that compares a low path typical sequence versus a high path consensus sequence, and you see an accumulation of basic amino acids that ultimately allow the virus to be establish a systemic infection in the birds. Okay, and luckily for us, those are only restricted to 2 subtypes. But again, we should not take the low pathogenic viruses as something mild in birds or inconsequential in birds. They do associate with significant losses in poultry as well. So, and with respect to the high path avian influenza viruses, we talk most of the time. These viruses were initially detected back in the mid-90s in geese in Guangdong, China. An important distinction between these viruses and all the other high path viruses that we have seen prior to this one is that this one, the first time it was identified, was already a highly pathogenic avian influenza virus. There is no ancestor of these viruses that show the typical low pathogenic phenotype, okay? So when they emerged for the first time, or the first time they were detected, they were already high path viruses. I think that at that time we had this realization that these viruses are a potential problem for humans because they did cause zoonotic infections. Until then, the dogma was that high path avian influenza viruses were really viruses restricted to poultry with no consequence to humans. Well, that concept changed in '97 when there were the first human infections detected in Hong Kong. 18 people were infected, 6 died, and a quick eradication of the virus in the Hong Kong area by essentially mass culling. The birds affected in the Hong Kong area led to basically dropping human infections to 0. But the viruses never went away. They remain in Asia and re-emerged with all their might, I would say, in Asia back in 2003. Not only that, the virus established back then a two-way transmission between poultry and wild birds that allowed the virus to spread to many other countries, causing not only problems in the wild bird population, but also in poultry outbreaks. And on the human side, the important thing is that it also was associated with human infections and high mortality of those human cases. The virus continues to evolve like many of these viruses do. They led to reassortment and eventually trans-Pacific migration that allowed the virus to enter into North America for the first time. That was around 2014 to 2016. The virus continued to circulate in Asia, again, continued to evolve into multiple clades, and those are refers as, a number. For those of you who are not familiar, there is a clade nomenclature that we follow, but the bottom line is that now these are not just a single virus evolving. There are multiple clades of these viruses in many parts of the world, and all of them have the potential to either become zoonotic or to cause disease in many animal species. And then we go all the way into the present time in which we have these viruses that essentially made it into the Americas again. But not only the Americas in North America, but the virus managed to go to Central South America and even to Antarctica, causing outbreaks in many different animal species. I want to highlight the outbreak in elephant seals in the south of South America, in Argentina. The mortality of and spread of this virus among elephant seals really affected the population, the animal population, and we are thinking that there's going to take many years for that number, the population, to recover from this outbreak. While at the same time, there is some indication that the virus remains in the area, causing outbreaks in different animal species. So in the United States, since December 2021, outbreaks in wild birds and mammals have been detected in about 14,000 detections in wild birds across all 50 states of the United States. There have been 647 reports of infections in mammals. I think the most important thing here, I mean, not to take away the importance in general that these viruses have, but the fact that this virus can infect domestic cats and cause disease. These animals live in direct contact with humans and could be that intermediate host that eventually leads to the emergence of a pandemic influenza strain in humans. But there are many different wild mammals that have been affected by these infections. And as I mentioned, migration of the virus to Central and South America, Subantarctica, mainland Antarctica in February 24, and the only part of the world that seems to have been spared from this virus is Oceania. But I think it's maybe a matter of time. In the case of the United States, the last detections you see that is widespread, very few in total number, but the virus is definitely widespread still in parts of the United States. And as I mentioned, one of the problems with this is the realization that these viruses are zoonotic viruses, and so far there have been cases associated with H5N1 infections around 900 or almost 1,000 cases. Eventually in every country that have had this virus for a while with an endemic nature have shown infections in humans. And in the case of poultry, it has severely affected in the United States the poultry production with over 170 million birds that have essentially been affected by these outbreaks. But we are not only at risk of H5N1 infections. As I mentioned, H5s are important, but there have been many other subtypes of avian influenza viruses. All of them, I would say, all of them have been associated with viruses that have become adapted to poultry and have led to these human infections, and some of them with very high mortality, as it was the case of the H7N9 infections that for several years affected mostly China between 2013 and 2017. That's where most of the infections occur. China took steps to essentially eradicate H7N9s from live bird markets, and by doing so, they have limited the spread of this virus and have led to basically no new human cases. I would say under the radar of many of these infections are the H9N2 infections in humans. These viruses are, as I mentioned earlier, very important because they are not only associated with economic losses in poultry, but because they have become adapted to the respiratory tract of poultry, their internal gene segments are very good at providing a zoonotic phenotype to other avian influenza viruses. And as I mentioned, H7N9s, H5N1s, those have been very important zoonotic viruses that inherited gene segments from these poultry-adapted H9N2 viruses. Serological studies suggest that exposure of humans to H9N2s is much higher than what the number of cases of H9N2 suggest. And in the past few years, we see kind of an increase in the number of cases in humans. Most of these cases are reported from China, but you could expect to have human cases in other countries where H9N2s remain endemic. So because of these viruses being an important problem, widespread around the world, I'm highlighting those that are in the Southeast Asia, Middle East, and Africa, as the most important viruses in terms of their problem for poultry, because those are where the poultry adapted H9N2s circulate. As you know, this is a very large area of the world. One thing that is, we're not going to be talking too much about it, but of all the avian influenza viruses that have caused infection in humans, these H9N2s from this part of the world have receptor specificity that most closely resemble what you would expect in a human influenza virus. So I would say of all the avian influenza virus circulate out there, the H9N2s are the closest to being kind of a human-like avian influenza virus. An interesting thing, sort of like a side note here, is that H9 subtypes tend to associate better with an N2 NA. Okay, so this is not a bias of just the poultry adaptation, but throughout the world, when you identify an H9, most of the time that H9 is associated with an N2 subtype. So there is some bias in the association of these two segments. So because H9N2s are a problem, many countries rely on the use of vaccines to control these viruses. An important aspect of H9N2s is that these viruses are not notifiable. So countries are not obligated to report the existence of H9s. So many countries rely on vaccination to control outbreaks of H9s, as opposed to culling of infected birds, because, you know, as it happens with H5s or H7s, that their presence may affect trade for specific countries. So in the case of H9s, a lot of countries rely on vaccination. Most of these vaccines are wholly inactivated vaccines with some adjuvant, all in water type of adjuvant, very common. These vaccines do provide protection, but that protection seems to be very limited. It produces, as the case of many inactivated vaccines, humoral immunity, but it provides no cellular immunity, does not provide mucosal immunity. These vaccines target the hemagglutinin, which is fine because it is the major target of the neutralizing antibody response. But I would say that it's very unidimensional because it doesn't take advantage of other antigens present in the vaccine that could also help with protection. And because it is a wholly inactivated product, it is not suited for mass vaccination. So essentially, you have to vaccinate one chicken or one bird at a time. One of the biggest problems that wholly inactivated vaccines have is the fact that influenza viruses are constantly changing. So this antigenic drift of HA makes these vaccines very ineffective. That leads to suboptimal vaccine or vaccination that can further promote antigenic drift on these viruses. Because of these issues, we thought about developing a modified live attenuated virus vaccine against H9N2s. There are many advantages of using a modified live virus vaccine. One of the advantages is that it mimics a natural infection, therefore it targets multiple antigens and can elicit mucosal cell-mediated in addition to humoral immunity. It has also the potential advantage of being a product that can be mass applied by either drinking water, aerosol, which I will be talking a little bit later, and in ovo, something we have not really tried with these vaccines very extensively. The disadvantages of a modified live virus vaccine. Well, the number one is always the fear of reassortment. Because these viruses do have a segmented genome, there is a chance that your components of your vaccine virus end up in a wild-type field strain, and that's something that is not something we would like to see. This is particularly problematic if we're talking about the hemagglutinin and the neuraminidase segments. We definitely don't want those segments to end up in a field strain, and this is very, very important to keep in mind. Like any other vaccine, the response will depend on the immune status of the host. Is that right? If it is too attenuated, or if the host has some level of immunosuppression, there's going to be lead to a suboptimal protection. But this is true for any type of vaccine. There is not something unique to ours. One thing that particularly developed countries that have poultry production very skewed towards poultry exports is that they would like to see the possibility to differentiate the infected from the vaccinated animals, something known as DIVA. In this case, we need to ask ourselves, is DIVA is really needed? Countries that do not produce poultry for export, but they have a flu problem, they may want to focus more on prevention and control of the problem than to just differentiate whether an animal was vaccinated or it was infected. So again, the poultry for export versus internal consumption is a big aspect of how countries need to decide the necessity of a DIVA strategy for the vaccine. And also there is the issue of being a vaccine against a notifiable virus versus a non-notifiable virus. In the case of the H9s, these are not notifiable, so it would be less of a problem, the use of a modified live virus vaccine. And if we use it against an H5, which is a notifiable disease, then again, will depend on the conditions of the country, how big the problem is, to decide whether a live product could be something of potential advantage to use in their respective country. So here in this paper, I encourage you to scan the QR code. It will take you to that paper. We show that we can generate a mass vaccination strategy with a modified live virus vaccines against H9N2 with a virus that is reassortment impaired, that can be delivered in the water and provided protection. I'm not going to go into all the details of the paper,
Okay, so I hope you got the concept of what we did with the hemagglutinin segment to decrease fitness and have a very specific marker. So the virus, this segment has less chances for reassortment. Something similar we did with DNA. We put something very specific, but in this case, the specificity is actually an immunomodulator. It contains the chicken IL-18 sequence. In addition, the virus contains two internal segments that have been modified, the segment 7 and segment 2. In this case, the second open reading frame in segment 7 that encodes for the proton pump M2 was moved downstream of the segment 2 PV1, downstream of PV1 in segment 2. And we put stop codons in M2 of the segment 7, so there is no M2 coming from segment 7, only from segment 2. Okay, and this is called, because we touched the M segment, we call this rearrange M or RAM strategy. Okay, we show, and like I said, I encourage you to scan the QR code, that these viruses are stable over multiple passages and they grow to good titers in vitro. Okay. Because this is published in the paper already, I'm not going to go into too much of the details, but to show the lack of reassortment of particularly the surface proteins that were modified, we did a co-infection study in which we put 10 times more of the modified live virus vaccine than the wild-type virus and did a co-infection in chickens and followed this infection over several days. Here an example of co-infection at 5 days post-inoculation from those samples. We sequenced these viruses by next generation sequencing, and we counted the reads that correspond to the vaccine or to the wild type strain. And you can see in blue here, particularly on the HA and the NA, that those reads are 0 and they are very high in construct for the wild type virus, indicating that the wild type virus has a biological advantage over the segments that were modified. And this is even in the presence of 10 times higher concentration of the vaccine compared to the wild type. So we have achieved something here that is very important, which is to reduce the fitness of both the HA and NA in the face of a wild-type virus infection. In addition, what we did was to infect chickens and put them in contact with direct contact chickens to monitor the transmission. I'm going to focus only on the bars in blue because that corresponds to the virus that has both the HA and NA modification. But again, we see by RT-qPCR viral RNA shedding over several days. But what you don't see is that direct contact shedding any of the viral RNA. So indicating that there was no transmission from the inoculated to the direct contacts. And the same situation was observed when we look at the clerical swabs. If you look at also in this graph, you will see that in general, the blue dots tend to have a lower share or lower amount of RNA compared to the black dots. The black dots correspond to the modified live viral vaccine with just the HA modification, not the NA modification. And to confirm that there was no transmission, we performed HIs with the sera from these chickens that were either inoculated or were put in contact. And you see, again, no evidence of transmission because the direct contacts did not seroconvert. But the ones that received the vaccine did seroconvert. And something that we have seen systematically is that despite the lower levels of viral RNA shedding from the virus that has the HA and NA modification, the amount of neutralizing titers tend to be higher with this virus than with the one that has only the NA, the HA modification. So, additional details are found in the paper. Here I want to highlight that we did drinking water vaccination of these birds. We started with 2-week-old Weiler horn chickens, followed by a boost. But again, we did two different strategies here. Prime only or prime boost, and we used two different doses, 10 to the 4 or 10 to the 6 EID50 per ml during vaccination. The graphs on the right show the stability of the vaccine at different temperatures, and you would see that the higher the dose, the higher the amount of virus that we can detect. And over the 2-hour period, the 10 to the 6 seems to be very stable at a wide range of temperatures from 25 degrees Celsius to 40 degrees Celsius. If you go higher than that, then you start losing stability of the virus in the water. I'm not going to go into all the details of this slide because there are a lot of graphs and a lot of information, so I will ask you to take my word for it. But the take-home message is the following. The protection, we could achieve sterilizing immunity, okay? But that was dependent on the amount of vaccine virus. So 10 to the 6 gave us that possibility of doing achieving sterilizing immunity and the number of doses. So the 10 to the 6 given twice was what led to sterilizing immunity. Everything else was sort of like in between with some chickens shedding some virus. But we, again, achieved very nice levels of protection with a high dose of vaccine given twice. And again, because that's published, I will leave it there. I will move into this more recent work that we have done. Can you hear me okay?
Yes, Dr. Perez.
Okay. And this has to do with sequential aerosol and oral immunization with a bivalent vaccine. Because again, we are interested in understanding whether our vaccine can also protect against H5, but without losing the perspective that H9s continue to be a problem, particularly in countries like Egypt that have both H9s and H5 circulating. So earlier in the year, we published on air liquid interface model for influenza aerosol exposure. This is something that we can do with in vitro tissue culture cells, but we can do it in vivo in ferrets with a nose-only aerosol exposure system that I'm not showing here. But we thought about using the same system to ask the question when we can either vaccinate chickens with an aerosol system. And I want to make a very big distinction between our aerosol vaccination versus what is core spray vaccination that is commonly used in poultry in the field. When we talk about core spray vaccination, we're talking about very big droplets, around 100 to 150 micron droplets, which are not necessarily very effective, although they do get into the respiratory tract of these birds. There are a lot of issues about standardizing these techniques, making a very good, even immunization with core spray vaccination. We thought about aerosol vaccination because those are with much smaller droplets, much more easily to go into each bird. So the ability to vaccinate would be better. But again, it's not used in poultry right now, and it will be needed some additional development on the engineering side to make this a reality for use in the field. But here we try to show that it's possible. So again, these chickens get exposed to an aerosol that basically delivers the vaccine. Here I'm showing the graph of virus shedding of chickens that received the modified virus with the HA and NA modification, this MLV H9N2 IL, or a bivalent H9N2, the same one as the monovalent, and an H5N2. We did not use an N1 NA here. And this is the group that was the prime only. I'll show you later what else we did. But here we're only showing virus shedding. And you can see that this virus shedding is very transient and can only be detected at 1 day post-vaccination, post-aerosol exposure. As a comparison, when we did drinking water vaccination in 2-week-old birds, we do see the virus until day 3 post-prime. So with aerosol vaccination, the way we did it, it's even more restricted, the number of days that the viruses can be detected. So here is the experimental design. Essentially, we took the modified live virus vaccine H9N2 monovalent in a prime boost, or a bivalent H9N2 and H5N2, either as a prime only or in a prime boost strategy. And we also use as a control a bivalent whole inactivated adjuvanted H9N2, H5N2 in a prime boost. I'll show you data. We did several challenges, a homologous challenge to the H9N2, but we also did a drifted challenge, a vaccine that does not, the virus does not cross-react with sera against the Egyptian strain. And also we did with the H5N1 a homologous challenge against this clade of virus 2.3.4.4b. The challenge with H9N2 is a very high dose challenge because chickens typically don't show much more, a lot of clinical signs when they get exposed to H9N2 in the laboratory. So we use 10 to the 8. This is a very large challenge. For the H5N1, we use 50 chicken lethal dose 50 to challenge. So this is what you get after prime and boost vaccination against the H9. So what we see after prime is the chickens that were vaccinated seroconvert. This is shown by the HI titers on the left. The monovalent, the bivalent basically had similar levels of HI responses. As we have seen previously, these HI responses tend to be lower than when you vaccinate with an inactivated vaccine. So this kind of makes sense. And post-boost, you'll only see the increase in the HI titers in the group that received the booster, not the one that did not receive the booster. And again, with inactivated vaccine, we see higher titers than with the live vaccine. The panel or the graph on the right shows the reactivity of the homologous virus from the vaccine, which is, again, HI titers that can be detected against the virus using the live vaccine or the inactivated vaccine. But if you look at on the left side of that graph, the drifted strain does not cross-react with the antibodies that were generated by our modified live virus vaccine. And there is just very limited, some level of cross-reactivity, not enough good HI titers when you use sera from the whole inactivated product control. But when we look at after challenge and consistent with what you would expect in the field, chickens that received inactivated vaccine, and I'm highlighting that with black arrows looking down, those are the groups that received inactivated vaccine, there was really not good protection, okay? The levels of shedding of virus are similar to the control that were not vaccinated. In contrast, any of the groups that received the H9 vaccine, either monovalent or bivalent, even the bivalent prime only, show no virus shedding. So we achieve again sterilizing immunity under these experimental conditions with a modified live virus vaccine, but you don't achieve that with a whole inactivated product, even though the whole inactivated product have higher HI titers. And when you look at in different tissues, in the sinuses of the birds that received the drifted challenge, have virus shedding. None of the chickens in the modified live groups had any shedding, indicating that our modified live virus vaccine is better at protecting chickens than a whole inactivated vaccine under these experimental conditions. What about H5? Well, here a couple of things to keep in mind. The protection, we did achieve protection. 9 out of the 10 chickens in the bivalent prime boost survived the challenge. And unfortunately, I cannot tell you that the one chicken that died here died because of H5N1. There were some logistical issues in BSL3 that prevented us from analyzing this particular chicken here. But in general, we got very good protection. And that protection depended on the regime. So prime boost was better than prime only, okay? And the presence of the H5HA antigen in the vaccine, because the monovalent H9N2 did not provide protection, good protection, although it delayed a little bit the time to death in these chickens. And you can see that type of protection and the shedding that you see consistent with what we saw with H9s. The one that was the bivalent prime boost tend to shed less virus than any of the other vaccine groups, even better than with the inactivated product. And in terms of tissues, a similar story by having the bivalent given twice, giving us the best performance, similar to the inactivated product in this case. And here again, the inactivated product may have worked better, but we're talking a very specific homologous challenge. It would be of interest for us to test in the future if we change with the drifted H5 high path strain, whether we can reproduce similar observations that we made with the H9 drifted variant. So how H5 protected? Well, I don't know how well it protected. One of the problems that we had with the H5 is that H5 of the Goose Guangdong lineage tend to be very poor antigens, all right? And when we look at HI titers against H9, this is the same graph that I showed you before, we see that those titers are very well detected. But when we measure with a modified live virus vaccine, there were no HI titers. So this is something that we have seen in the past, not only in chickens, but in ferrets too. So these modified live virus vaccines don't seem to elicit a very good response. However, they do elicit a response that is specific to H5 because otherwise we get no protection. And the inactivated product in this case also led to very poor responses. To try to understand this a little bit better, in the lab we use a neutralization test that is based on nanoluciferase. These are viruses that express nanoluciferase, and the curve shows essentially the level of antibodies. So if I look at the H9, the inactivated vaccine, very nice titers, consistent with the HI titers, the bivalent prime boost also, etc. And the monovalent prime boost, very good titers. The bivalent given once has the lowest titers. When we look at the H5, we know this is a specific. The inactivated gave us some titers. The bivalent given twice gave us a little bit of titer. The bivalent prime only has some level, and the H9 does not cross-react. So we do detect some antibodies with the anti-H5 in this case. And by ELISA, we can detect, again, very nice titers against H9, very poor responses against the H5. Only after boost we detect some responses. And the H, obviously, we see it with inactivated product. We do have we detect N2 responses, particularly after boost, something that we don't detect against N1 and NA, which makes sense because these viruses only contain an N2. So you can loosely say that, well, we have sort of like a diva strategy if the H5 is circulating as an N1 or an N8. But the problem here is that then if the virus decides to reassort with an N2 subtype, then our diva strategy will not be that useful. Okay, so I said in my slide at the beginning that I was going to talk about cows, but I'm not going to talk about cows. I'm going to talk about the need to faster responses to avian influenza viruses with the help from in-farm diagnostic tests. So we were approached by a company, Alvio, that has this technology, AlvioSense detection system. This is a very user-friendly. You don't need people to have a lot of training to use this kit. Essentially is something that has a small cartridge where you're going to put your sample in. You put the sample into this little device here, and this device connects via Bluetooth with your phone, and it can tell you whether you have flu or not in the sample. This cartridge will, in this case, this cartridge was designed to detect type A influenza, subtype H5 or subtype H9, okay? And we decided to give this a try to see how these samples will behave in the context of a flu infection on something that resembles, a field condition. And more importantly, how we work in duck samples, because something that the company did not have was good information about samples from ducks and potential inhibitors in the sample that would affect the detection of the virus in the sample. So we decided to use a highly pathogenic H5N1 in ducks. We did 2 experiments with this highly pathogenic virus, and also we did a low-path version of this virus. This is essentially the same virus as in the left, but the HA has been modified to be of low path, okay? It doesn't cause more. We run the Alvio system here using cloacal swabs. Bottom line, and I will go very briefly because I think I'm speaking longer than I should. We saw that this Alvio signal was very good, very specific, very consistent. Even by day 5, cloacal swabs, we had only one duck was positive by TCID50. That was enough to give us a good signal for H5 infection, okay? When we go to the low-path version of this virus, number one thing, the low-path version of the virus replicates better in ducks than in chickens. This is consistent with the notion that these viruses are becoming more adapted to wild birds than to poultry. These are viruses that circulate in North America. These are not your typical Asian H5N1s. And the virus showed that in chicken oropharyngeal swabs, very nice detection of type A influenza and H5. And in ducks, we saw that both on day 4, for instance, the oropharyngeal swab and the cloacal swabs were positive. And in general, we have some minor issues. This one that says invalid is basically a problem with the sample that we run into. That's something that I don't know how common it's going to be, but in general, I would say that this is a very reliable system for detecting early in the infection the presence of virus. More recently, we did an experiment in which viruses were challenged with a very low dose of H5N1 high path. And by day 1 post challenge, we do get a very nice detection of the virus with the Alvio system, but not with another antigen capture system, which has less sensitivity that resulted in negative results. So the Alvio system was better at that in that case. So in summary, I told you about modified live virus vaccines, an aerosol vaccination strategy that might be expanded into something that we can actually apply in the field. We developed a vaccine that is bivalent that provided good protection against H5 despite very negligible anti-H5 antibody responses. And more importantly, I think I could convince you, hopefully, that even in the presence of a drifted H9 challenge, the live vaccine was superior in protection compared to the whole inactivated vaccine control. And in the case of the diagnostic system, with very nice currently identification of 100% of ducks that were challenged with the high path virus. With the low path version, there was, in general, very good detection, but there were some potential days in which the system did not work as you would expect based on the amount of virus that we used to infect. But in general, I would say that this is a very powerful kit to detect early in the infection avian influenza infections in chickens and ducks. One of the problems with the avian flu rapid diagnostics, and perhaps something that you can discuss among yourselves, is this need to have a more globally harmonized data on how well these rapid tests work. I mean, I just show you one of them from one company. We like it, but it doesn't mean that there are not others that are better or that could complement some of these things. So I think it's important we have discussions to try to benchmark this rapid test, particularly the lateral flow tests that detect antigen. They tend to be less sensitive than something that is based on an amplification step. So there is some talks about developing a collaborative network that can do this better, and I think that the more people interested in joining this type of initiatives, the better, because it will essentially lead to more confidence into these different kits and how well they behave in real life conditions. So with that, I think I'm done. I don't know. I'm happy to take any questions if there is any time. But I hope it was useful.
Okay, thank you very much, Professor Dr. Daniel Perez, for such a wonderful presentation. And we are deeply grateful for your time and for your such a wonderful presentations. So we will now move into a Q&A session. I would like to invite our co-moderator, Mr. Umair Asgar, who is the PhD scholar at Rockland Environmental Sciences University, Poland, and the Secretary General of the PPS. So it's over to you, Umair Asgar. Meanwhile, while Umair, you collecting the questions from the audience side, Dr. Perez, I have a question from your presentation. With the virus now established in a global wild bird reservoir, is the goal of eradication no longer feasible? Should the global strategy now shift entirely towards long-term management using a combination of vaccination, biosecurity, and surveillance to protect poultry, economic interest, and ultimately human health? So what is it?
So eradication from wildlife is, impossible, but working towards eradication out of poultry, I think is still feasible. Is that right? It's how you control the flow of contact between wildlife and your animal production systems. So, one of the things that we need to start addressing, and I've said this, sometimes it's easier to be said than done, but one of the problems that we have is with animal husbandry in general in many countries. So we tend to raise animals the same way for the past, I don't know, 500 years, 1,000 years, I don't know. And I think we need to start thinking about what is the best approach, because to start changing animal production systems little by little So we reduce the chances of introducing stuff. I mean, biosecurity, I mean, this is an emphasis. We emphasize biosecurity all the time, is that right? But these barriers that prevent or are designed to prevent something from coming in or from going out, for that matter, are very loose, are very porous. Is that right? They're not very good. So I mean, I've seen places in which some of the biosecurity measures are being taken in consideration, follow a handbook from a different area. So I've seen pictures from people in some countries in Asia that they're following something that probably they would do in Europe, and the situation is not the same. You have some countries that have 50 Celsius degrees weather, and you are asking people to go with Tyvek suits or plastic suits and try to not bring something in. But then when you look at the production system, a lot of things can go in that is not human. Is that right? So so we need to start changing and addressing maybe on a case-by-case basis what steps we can take to minimize things from coming in, and if they're in, what steps we take so things don't go out. Is that right? So that I mean, that's in addition to the potential use of vaccines as something that could be useful. Is that right? Because vaccination without an additional plans on how else we're going to do to prevent viruses from coming in or going out if they get in, it's useless. Vaccination is going to be useless. Is that right? And it can end up doing more harm than good.
And how surveillance plays a role in this line?
Very important. If you're going to use vaccination, you have to have very strong and reliable surveillance systems. You need to monitor very extensively the presence of virus or other flu viruses. Is that right? So we have examples. We take care of the H7N9 problem, but there is an H3N8 that also manages to go to humans. So we need to have better systems for monitoring. And those better systems for monitoring doesn't need to be, I mean, again, depends on the situation and the resources. Is that right? But vaccination without a proper surveillance system, I think, is going to be a failure. So
Thank you very much.
if you decide to vaccinate, you need to include surveillance in the equation.
Thank you very much, Dr. Perez. I'm clear with your answer. So now it's over to you, Umer.
Okay. Can you hear me now?
Okay.
Okay.
Yeah.
Thank you very much, Professor Perez, for such an insightful presentation on innovations in poultry vaccination, live attenuated strategies against bird flu. On behalf of PPS Society, we deeply appreciate your sharing your expertise on this critical subject. We will now begin our Q&A session. We have received a number of thoughtful questions from our participants. Professor Perez, are you ready?
Yes, I am. Thank you. Well, I'm as ready as I can be.
Thank you. So our first question is from Mr. Sajjad, who is asking what's the difference between influenza virus and Binna virus? And two at postmortem level at bronchi bifurcational point, please.
Umer, can you please repeat your question?
Yeah, sure, I will. Mr. Sajjad is asking about the difference between influenza virus and Binna virus.
So completely different virus families. Okay, so they have no relationship. So yeah, I would not pretend that I know
I
much about
think
birnaviruses.
there was a second part of his question, Sajjad Hussain. He was asking about post-mortem level at bronchi or bifurcational point, please. Matlab, I think he want to clear a difference between Birna and influenza virus in terms of post-mortem lesions.
I would not be able to comment on that because I don't do a lot of work with birnaviruses, so I would not, yeah, be able to comment on that.
May I
Okay,
move
so
on to the next question,
yeah,
please?
so we can move further to our second question again from Mr. Sajjad, who is asking about the please briefly share the recommended dose, water quality needs, and the best age and booster timing for course spray vaccination.
I'm not sure that I follow. Sorry, sorry.
Actually,
I,
Professor, he's asking about the water quality, dose,
Oh,
and the best age
the water
for
quality.
vaccination.
So yeah, I mean, definitely, we treat the water so the chlorine in the water would not inactivate the virus. So that there is a procedure that we follow to, and we in the papers that we publish, I think if I'm not mistaken, we describe what the procedure is. But certainly, the quality of the water, how you treat the water prior to making your virus dilution is important, and we took care of that. So, additional part of that question was,
The additional point is about the booster dose,
oh, the
booster
booster
timing.
dose. The booster
Mm-hmm.
dose and booster timing. So the booster dose
Yeah.
is the same as the initial dose. So we tend to favor 10 to the 6 egg infectious dose 50 per dose. In the case of the drinking water, that was 10 to the 6 EID 50 per ml. And we calculated that every chick drank about 4 mLs of water in the time span that we put him in there. It's very difficult to do it on a per chicken basis. That's a calculation based on the amount of water that we saw the chickens consume over the 2 hour time that we put it in there, the water in there. To do the vaccination in the way we did it, we removed the water essentially for 12 hours and put it back in with the with the water that contains the virus. And in terms of the timing, we typically do our prime boost strategies 2 weeks apart. So we do the prime, and 2 weeks later we boost them. Again, we tend to use the same dose for prime and boost.
Got it. Thank you very much, Professor, for that clear explanation. So let's move to our next question, again from Mr. Sajjad. He's asking about the recombinant vector vaccine.
Which one specifically?
I think, Matt, these are the questions that we can
Yeah,
just Google it,
we can
or
see, we can go more further.
if there
Okay,
is some technical
so
questions.
okay, sorry for that. So Dr. Said is asking about what's the take-home message?
The take-home message is
Yeah.
that modified live virus vaccines should be given a shot. I think that we've done the homework to show that we can limit the reassortment potential of a live virus like influenza, which has always been, or it continues to be, a major issue. I think we've taken steps towards alleviating that concern. And our data clearly shows that at least for the H9, we got much better, broader protection than with an inactivated product. And you asked a question about recombinant vaccines. I think that recombinant vaccines can be powerful tools. The advantage is that you're not talking about a live virus like influenza, is that right? But they tend to be also very unidimensional because these recombinant vaccines usually only incorporate the hemagglutinin into it and try to elicit an immune response. We are going to do soon, hopefully soon, a comparison of the protection of the level of protection that we achieve by either aerosol administration or drinking water administration of our modified live and a Newcastle disease virus recombinant that encodes the same H9HA. So we're going to do a side-by-side comparison to see if there is any advantage of our vaccine versus a recombinant vector like NDV. There are other, vectors like HVT, herpes virus of Turkey, recombinant vaccines that can be applied in novo, has the possibility of doing in novo vaccination. The problem with that vaccine is like many herpes virus vaccines, is that to elicit a good immune response, you need at least 30 days. Is that right? So, as you know, many countries in the Middle East, they prefer their chickens young. So by day 28, they're already on a table. They're no longer alive. So if you have a vaccine that it takes a month to elicit an immune response, that's not going to help you. So I think we overcome some of the limitations with the technology that we are developing.
So thank you so much, Professor, for such a nice explanation about this question. So the next question again from Dr. Said. He's asking about the modified live vaccine for H9 is recommended in broilers, in your opinion?
So that's the kind of response that people in the respective countries and regulatory agencies will have to make. I mean, it's their call. I think it could be used in broilers. I think it could be used in layers. I think it could be used. I mean, we don't see a problem of using these in different type of birds. But again, the decision where this is going to be used depends on what countries would be allowed to do.
Got it. So our next question is from my side, Professor, if I can.
Yeah, sure.
Yeah. So I have a question regarding vaccine delivery methods. Professor, from a practical standpoint, which delivery methods for live attenuated vaccines have shown the best balance between efficacy or in terms of, you can say, the administration in large commercial flocks?
So one thing is what people do typically in the field and what we are trying to do with our approaches. Is right, as I mentioned earlier, aerosol vaccination is not used in the field. People use spray vaccination. And spray vaccination, you need to train people very well on how to do it properly. Oftentimes you see not being the failure is not on the vaccine itself, but in the way it was applied. All right. So but it's prone to a lot of spray vaccination is prone to a lot of operator issues more than the vaccine itself. Is that right? So I think that's not a very efficient system. I mean, it works, right? I don't want to take anything away from the fact that it does work, but it works when you do it properly. Is that right? I think that if we were to explore a little bit more this aerosol type of chamber system that I show you, we will have a much better way of vaccinating chickens with a more homogeneous delivery of the vaccine, because we're talking about much smaller particles. These smaller particles will tend to go deeper into the lung of the birds, and it will tend to elicit a more complete immune response. Okay. But the standard in the field is either you use one bird at a time or you use some form of spray vaccination, where this is gel-based or a spray. It's still the standard in the field. I see a lot of issues with that in the terms of how different people apply it.
Thank you so much, Professor.
Oh, and you asked me about what would be the best strategy. So the best strategy
Yes.
to me would be, if we can develop the aerosol system, I would say aerosol at the hatchery. It should be something that can be applied. So if you have a hatchery, that's where you do the aerosol vaccination. And then if you need to do a boost, you do drinking water vaccination like we did in the experiments.
Okay, so thank you so much, Professor, for your detailed answer to this question. So this concludes our Q&A session. A huge thank you once again to Professor Dr. Daniel Perez for his fantastic presentation and for sharing your profound knowledge with us. It was a truly valuable session with you, Professor. Thank you so much on behalf of PPS Society. And now the floor is yours to Mr. Sayam again. Thank you so much.
Thank you very much for co-moderating the session Q&A, Dr. Umair Asghar. One of the last question from my side, Dr. Daniel Perez. I think we are taking a lot of time. Regarding the LVO system, your presentation mentioned regarding the field diagnostic, right? So how would you recommend that system and how effective this system is? And is it globally, in your opinion, and is it globally recognized or launched or not?
So the company, so I cannot speak on behalf of the company. I don't work for the company, but I understand that the company is trying to get approval in different countries, including the US, the UK, and it was initially developed by folks in the Netherlands, in the Royal JD company. So those guys were initially involved in the development of this. This company initially developed a lot of similar technology for COVID. So they came from the human side and going into the poultry side. So I think they're still learning the ropes of how to get approvals and things like that. So I would say it's not widespread use, but the hope is that it will get approval in different countries and can be, I think it's a reliable system. My modest opinion, and I have shared this with the company, is that maybe the cost is an issue. Okay, the cost of using this test is going to be an issue, particularly in the poultry side. But if they can work out the cost issues, I think that this is a very nice complementary system to any good diagnostic lab system. Is that right? So the only concern with this system sometimes is that people will find something and not communicate something. So that's an issue. But again, I cannot talk for what other people in other countries or what people in this country would do if they have a system like that. I think that in the US, the conditions are such that would favor reporting, so people will not hide a diagnostic test, a positive. In other countries, I don't know what the situation would look like, but I think it's a very valuable tool, and people should. I would encourage reach out to the company, reach out to Alveo Technologies, and ask them, what they can do to essentially incorporate their system into whatever strategy you think that would work, for instance, in Pakistan. I don't know. I mean, it's just a suggestion. Again, I don't work for the company, okay? I'm trying to help them with establishing whether the system works or not. But again, I don't want you to take my words as I'm pushing Alveo's technology. I'm pushing any technology, any rapid test that works. Is that right? So it's not just Alveo. I think we need to start thinking of this test as something that can complement our diagnostic systems.
Okay, thank you very much, Dr. Daniel Perez, for your such a wonderful presentation and the Q&A session. You covered all the area. So now